Hello and welcome everyone. So in this particular video today we'll discuss about the iron sulfur proteins. The iron sulfur proteins basically they are characterized by iron sulfur clusters that containing basically they are characterized as iron sulfur clusters. They are further characterized by sulfide bridge. They contain what the sulfide linkage or sulfide bridge in which there are di, tri, tetra iron center is there with the different variable oxidation state. So basically that iron which is exist in most of the biological participate in biological activity. Let's say example redox reactions in hemoglobin, in cytochrome, in myoglobin, etc. The iron sulfur proteins are responsible for the electron transfer phenomena. They are specially used in the biological activity and they perform the function as electron transfer. There are certain examples are ferrodoxin. One more example that is also worked as a catalysis. They are used in the biocatalyst. Let's say example nitrogenase enzyme. Let's say example NADH hydrogenase. Let's say hydrogenase. Okay, basically NADH dehydrogenase. Dehydrogenase. Hydrogenase and the example is nitrogenase, etc. The iron sulfur clusters are best for their role in oxidation reactions, particularly in the mitochondrial electron transport processes now. So they are worked as electron transfer, they used in the electron transfer phenomena. They also regulate some gene functions. What iron sulfur proteins are regulate the gene expressions, one can say. The iron sulfur proteins are used in particularly for biogenic nitric oxide attack. And in most of the iron sulfur protein, the clusters perform the best function is electron transfer. That's why I highlighted here the electron transfer phenomena is the basic activity of the iron sulfur protein. The iron sulfur proteins are classified on the basis of their structures. Let's say the number of iron which are present in that. The first example which is called as a rubredoxin. The first example which is called as what? The rubredoxin. Now this rubredoxin in which the active site is an iron, in which what? The active site is an iron, and particularly it is a low molecular weight iron that which is particularly found in the bacteria which is involved in the electron transfer reactions, okay, which is transferred in the what? Electron transfer phenomena, okay. It is also basically the kind of what iron sulfur protein, okay, and it consists of it have the tetrahedral geometry. Now look at the structure of that iron which is exist in the rubredoxin. Here this is the tetrahedral arrangement, okay, and these are the sulfur which is from the cysteine residue of the amino acid, okay. This is what the tetrahedral arrangement now, and this is the active core is an iron. So rubredoxin which contain iron either in the 2 or +3 +2 or +3 oxidation state, and which is coordinated by 4 sulfur from the cysteine residue and which form the tetrahedral arrangement. So the rubredoxin is one electron transfer species, or that is performing the one electron transfer system, okay. Here the iron from +2 is going to convert into what? The +3. So that is what? The one electron transfer phenomena. So the iron atom changes their oxidation state from +2 to +3, and in both the oxidation state the metal exists in the high spin state now, okay. Metal exists in the what? High spin state now, and which helps to minimize the structural changes now. So the oxidized form is reddish in color. If you see the oxidized form, that is Fe tripositive, which is the reddish in color, while the reduced form which is colorless now. which is what the colorless, because at the electron transition has the transition which is done in the IR level, okay, that is called the infrared level, okay, and which is cannot seen by what naked eyes. So this is about the rubredoxine. Now the next one which is called as the ferredoxine, now which is called as what? Ferredoxine. Let's see about the chemistry behind the ferredoxine. The second one will be called as a ferredoxine. Now this ferredoxine is the first example for the ferredoxine for is 2 iron 2 sulfur type of the protein now, okay, 2 iron 2 sulfur, and structurally it is bitetrahedral geometry now. So what is that bitetrahedral geometry? So that bitetrahedral geometry means that each iron, okay, is in the tetrahedral arrangement, okay, and look at the structure for ferredoxine. Here this is what one iron, this is the second iron, okay, and these are the, this is what the sulfide bridge, okay, look at the structure of the, this is what the sulfur, and these are what the sulfur from cysteine residue, which is an example of what, or which is a part of what, amino acid, okay, look at this sulfur. And here in case of what ferredoxine, there are 2 sulfide bridge are there now, there are what, 2 sulfide bridge are there, okay, and again it is worked as a electron transfer phenomena, okay, it is for what, the electron transfer phenomena, which is used in the electron transfer phenomena, okay. One more example for the iron sulfur protein, that is called as a so the another example that is called as a the rescue protein now. So rescue protein is a type of what, again the 2 iron containing system. So here that is called as a rescue protein. So here the name as a rescue protein. And in the case of what rescue protein, so structurally it is, again it is a tetrahedral bitetrahedral geometry where 2 irons are there, and which is linked with the 2 sulfide bridge, and here the nitrogen is there, one of the iron which coordinated with the nitrogen, 2 nitrogen atom from histidine, and here the 2 coordination side from what, the sulfur. Here this is called as a Fe2S2 type of the iron cluster, Fe2S2 type of the iron cluster, in which the one iron is coordinated with the 2 sulfide bridge and 2 cysteine, and similarly other is also with 2 sulfide linkage and the 2N histidine now, which is involved in the electron transfer reaction, in particularly for what, the mitochondria, that is electron transfer in what, the respiration process in what, the mitochondria, and photosynthesis in the chloroplast. So basically the use is electron transfer, and in the two way it do the electron transfer reactions, first in mitochondria, that is for respiration, and in chloroplast for the photosynthesis. So these are the uses and particularly the structure of rescue protein, okay. So this is about the sulfur iron sulfur protein now. Now one more thing is what, the last structure, that is called as a the ferredoxine cubane like structure, ferredoxine cubane like structure. Now what is that cubane like structure? So where there are 4 iron linked with the 4 sulfur, 4 iron with 4 sulfur, and they are mentioned as if 4 Fe4S4. Now the structure is like a cubane. So what is the cubane now? Look at the structure of cubane. This is what the cubane, okay, and here the irons are, this is what the iron which are present diagonally, this is what the 4 iron. and now remaining are what? The sulfur, 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 sulfur and sulfur. Okay, and now that each iron is coordinated with the cysteine. Okay, this is what the structure of the ferrodoxine Fe4S4. Again, it is participate in the electron transfer mechanism, and there are two types of what particularly the ferrodoxines are there, that is called as a high potential ferrodoxine and the low potential ferrodoxine, where high potential in the which is in the positive, okay, with the positive number, and low potential means in the which is in the negative. Okay, that's called the low potential. And again, it is participate in the redox reaction. So in oxidized form, maximum number of iron are present in the tri-positive state, and in the reduced form particularly the maximum numbers are present in the di-positive. So this is all about the iron sulfur protein, which are actively participate in the bacterial, one can say, in the bacteria for the purpose is what? The electron transfer mechanism. So thank you for watching this.